Hello everyone. Efruzhu cancer carcinogenesis theory and molecular mechanisms, volume 4, third part, molecular carcinogenesis, EFRUZ ASİLOÐLU, independent researcher. Copper is the third most abundant essential trace mineral in the body after iron and zinc. Most people consider it unimportant. Much has been learned about the important biological roles copper and copper-dependent enzymes. Copper has an entirely different role in the body, being a component of two of our most important antioxidant enzymes, copper, zinc, superoxide dismutase, and ceruloplasmin. Unbound free copper is not found in large quantities in the human body. Almost all of the copper in our bodies is bound to either transport proteins, ceruloplasmin and copper albumin, storage proteins, metallothioneins, or copper-containing enzymes. Copper is essential for the proper functioning of these copper-dependent enzymes, including cytochrome C oxidase, energy production, superoxide dismutase, antioxidant, protection, tyrosinase, pigmentation, dopamine hydroxylase, catecholamine production, lysyl oxidase, collagen and elastin formation, clotting factor 5, blood clotting, and ceruloplasmin, antioxidant protection, iron metabolism, and copper transport. The adult human body contains between 80 and 150 milligrams of copper. The National Research Council has established a tentative, safe, and adequate daily intake for copper, which for adults is 2-3 milligrams daily. Vitamin C or zinc, you should pay close attention. This is because convincing evidence has accumulated suggesting that zinc and vitamin C supplements are strong antagonists of copper status. Copper's role in cardiovascular disease, diabetes, arthritis, osteoporosis, free radical damage, cancer, inflammatory disease, immune function, blood lipids, and thyroid function. Copper deficiency can significantly increase the plasma cholesterol concentration. Additionally, this increase in cholesterol results in an increase in LDL cholesterol and a decrease in HDL cholesterol. Copper deficiency significantly increases the susceptibility of lipoproteins and cardiovascular tissue to lipid peroxidation. The function of copper as an integral component of copper zinc superoxide dismutase and ceruloplasmin is well established. Copper zinc superoxide dismutase, for example, performs antioxidant function in varied tissues and fluids and is indispensable to oxygen metabolizing organisms. Copper deficiency has been shown to result in a twofold increase in the level of lipid hydroperoxides in liver mitochondria. Adequate copper is vital for optimal functioning of many antioxidant enzymes, both copper-dependent and otherwise, in varied organs and tissues. It has been shown that functioning of macrophages were decreased in severely copper-deficient rats. But even marginally copper-deficient rats had impaired immune functioning, neutrophil superoxide dismutase activity, and neutrophil function was significantly diminished, suggesting that immune function may be more sensitive to diets low in copper than standard measures of copper status. Copper is serum level elevated in animals and humans with cancer, often like the elevation of serum copper in cardiovascular disease. It seems that the elevation of serum copper that occurs in conjugation with cancer is part of the body's biological response to the cancer rather than its cause. Numerous studies examining varied types of tumors have demonstrated that with remission usually comes a decrease in serum copper levels to normal. Interestingly, most tumor cells have decreased copper zinc superoxide dismutase activity compared to normal cell, and it has been suggested that the elevation in serum copper is physiological response designed to activate superoxide dismutase or other copper enzymes in cancer cells to inhibit their growth. Indeed, numerous copper complexes that demonstrate superoxide dismutase mimetic properties, including copper salicylate, have been shown to possess anti-cancer, anti-carcinogenic, and anti-mutagenic effects, both in vitro and in vivo. In fact, there is some experimental evidence that copper complexes can cause established tumor cells to differentiate into normal cells. Copper is an essential component of several endogenous antioxidant enzymes, and free radicals have been proposed to play a role in the process of carcinogenesis. The effects of dietary copper levels on the development of cancer has been investigated. Rats fed low copper diet show a higher incidence of carcinogen-induced colon tumors when compared with rats fed a high copper diet. The development of anti-inflammatory steroids and aspirin-like non-steroidal anti-inflammatory drugs quickly replaced copper compounds in the treatment of these conditions. Copper-containing enzyme ceruloplasmin is significantly elevated in inflammatory conditions and has anti-inflammatory activity. Has been expanded to include, in addition to inflammatory disease, gastrointestinal ulcer, cancers, carcinogenesis, and diabetes. In these conditions, much of the research interest has centered on the finding that many copper complexes demonstrate superoxide dismutase activity. Because of this, many of these compounds have been designated as superoxide dismutase mimetics. Most human supplements of copper contain either copper sulfate or copper gluconate, two well-utilized forms of copper. However, because copper outside of biological systems can catalyze oxidation, total copper supplementation should not exceed 5 mg daily. The chemical symbol Cu and the word copper were derived from Roman name for the Cyprian metal, cuprum, because their source of copper was Cyprus. In 1931, it was discovered that copper was more effective than iron for hemoglobin formation. We now know that it is an essential element necessary in the diet since it can't be manufactured by the body. Copper is necessary for the conversion of iron to hemoglobin, necessary in improving energy and alertness. It also assists in the utilization of vitamin C, reaching the bloodstream within 15 minutes after ingestion. It assists amino acid tyrosine to work as a pigmenting factor for skin and hair when bound to such substances as zinc and ceruloplasmin. Copper becomes an important blood antioxidant, preventing polyunsaturated fats from turning rancid in the body, thereby limiting the production of free radicals. Ceruloplasmin is an acute phase reactant or an emergency antioxidant that can't function without copper. In testing, copper has protected against viral and chemically induced cancers. Although not easily destroyed, copper is required only in small amounts, with the rest being excreted. Copper also exists in an unbound form known as copper ions. These ions can be powerful generators of free radicals. It has been suggested that the contraceptive loop worked on this principle, killing sperm by its generation of oxygen radicals. Copper deficiency are thought to be responsible for anemia, low white blood cells. Copper deficiency in young men leads to a significant lowering of HDL in the blood. Copper is involved in carcinogenesis not by direct mechanism but by antagonizing selenium, thereby diminishing selenium protective effects against cancer. Researchers have found elevated copper levels in certain kinds of cancer, but further research is needed in this area. Copper deficiency symptoms: anemia, red blood cell rupture, abnormal skin, hair pigmentation, difficulty in breathing, etc. Copper can be detoxified by manganese, molybdenum, selenium, and zinc. Copper and cancer. Copper is a trace element found naturally in foods such as seafood, organ meats, green vegetables, and nuts. It assists in the regulation of blood pressure and heart rate and the absorption of iron in the body. Studies have found that copper has antioxidant properties and may have some anti-cancer effects. Other animal studies suggest that high copper levels may promote cancer formation and growth. Fruits and vegetables can contribute up to 30% of person's total copper intake. The estimated minimum daily requirement is 2 milligrams. Animal studies have shown that copper is useful in maintaining antioxidant defenses. Antioxidant compounds block the action of activated oxygen molecules known as free radicals that can damage cells. While the involvement of copper in the cancer process via antioxidant effects is still unclear. Copper complexes have been shown to have anti-cancer properties in laboratory studies. Other laboratory and animal studies suggest that high copper levels may promote formation of liver cancer and stimulate growth of brain tumors. Copper is a necessary nutrient for absorption of iron into the body. Cancer-fighting minerals. Beneficial compounds in food and supplements may intervene along a line that runs from initial exposure to carcinogens to the final step into outright malignancy. Nutrients can counteract an environmental poisons and the toxic byproducts of liver metabolism neutralize free radicals which might otherwise cause carcinogenic mutations in DNA, boost the immune system, inhibit enzymes that drive cell proliferation, halt metastasis, cancerous reproduction. Dietary guidelines advocated by the American Cancer Society and the National Cancer Institute suggest eating plenty of fruits and vegetables, get lots of fiber and limit fat, especially animal fat, red and orange pigments found in fruits and vegetables. Carotenoids supply free radical fighting activity. Carotenoids found primarily in tomatoes supply double the free radical fighting activity of beta-carotene, the most widely studied carotenoid. This carotenoid may be the best at lowering a man's risk of prostate cancer. Resveratrol, a constituent of the skin of red grapes, is found to protect hearts by blocking oxidation of LDL cholesterol and discouraging blood clotting. Now tests show it also squelched both free radicals and inflammation, two well-known cancer inducers. copper and zinc salicylates. Sometimes there is too much of the inorganic form in drinking water. Acid water flowing through copper pipes, it can then gradually accumulate in the body, lead to toxicity symptoms. With signs of zinc deficiency, high copper water in animals helps to keep them free of parasites. Copper armbands are well known to reduce arthritis. Copper serum levels are elevated up to 3-fold above normal with inflammation and with many chronic and infectious diseases, apparently because the body mobilizes all tissue store of copper to fight the condition. During remissions, the copper level returns back to normal. The most effective anti-inflammatory agents are copper complex. Commonly, these are related to salicylic acid. The copper enzyme is also needed for the synthesis of adrenaline, epinephrine. The main metabolic defect of cancer cells, according to researchers, is the deficiency of enzyme cytochrome oxidase. This causes a blockage in the cellular respiration or oxidative energy production of the affected cells. Dr. Budwig claims that plenty of linseed oil and sulfur amino acid, L-cysteine, and especially L-methionine help to correct this situation. Cytochrome oxidase is a copper-dependent enzyme, and additional copper might be of further benefit in the final stages of this oxidative energy production. Electrons are transferred to copper 2 and iron 3 in the cytochrome oxidase to form copper 1 and iron 2. In the last step, these electrons are then transferred to oxygen, which now can attract hydrogen ions to form water. In cancer cells, this transfer of electrons to oxygen is blocked and energy is being produced very inefficiently by converting glucose into lactic acid. However, practical experience has shown that copper is more important to prevent cancer or regrowth of tumors and possibly with dormant tumors. If growing tumors are present, then copper is needed to form new blood vessels. Taking high amount of zinc creates a relative copper deficiency and helps to prevent the formation of new blood vessels. Copper salicylates have a similar actions as superoxide dismutase to protect cells from free radicals. The therapeutic potency and safety of the copper complexes of aspirin, acetyl salicylic acid, and salicylic acid is much better than for aspirin itself or for inorganic copper. These complexes are 5 to 8 times more effective than aspirin, but less toxic. Harmful effects of aspirin salicylic acid and similar drugs apparently arise because they bind copper in the body and cause a localized copper deficiency in the tissues. Copper 1 salicylate is a strong antioxidant and also appears to have good anti-inflammatory qualities, but all the published papers are on copper 2 salicylate. However, the only data for a copper 1 complex that could find penicillamine showed it to have even stronger anti-inflammatory activity than the corresponding copper 2 complex. The only problem is that ascorbic acid easily becomes oxidized in contact with metal. Therefore, it is best to exclude all air. Zinc has strong anti-inflammatory and antibiotic properties as well as, but can become deficient with high copper intake. Therefore, it is usually best to increase the intake of both minerals together. The chelating effect of salicylic acid, which may cause zinc and copper deficiency in the body. Therefore, in the form of zinc and copper complexes, salicylate may not normally cause a reaction in susceptible individuals. Animal thioredoxin. Animal cells contain a classical thioredoxin system, including a single type of thioredoxin and thioredoxin reductase, which uses NADPH as an electron donor. Although mammalian thioredoxin appear to target proteins and display functions that differ from one tissue to another, most, if not all, are involved in co- and or post-translational processes. This prominent feature suggests that thioredoxin targets proteins of general importance to cell growth and multiplication. Thioredoxin can act as a radical scavenger under oxidative treatments known to generate reactive oxygen intermediates. Intracellularly, UV, X-ray irradiation, or exposure to hydrogen peroxide, thioredoxin is effective of facilitating the regeneration of oxidatively damaged proteins. A series of dilation analyses show that thioredoxin expression is enhanced through a cis-acting promoter element responsive for the oxidative stress. Recently, thioredoxin has been localized in the pregnant human uterus, where it may protect the fertilized egg and placental trophoblast from the cytotoxic effect of oxygen radicals. The effectiveness of thioredoxin as a radical scavenger has also been recognized through its ability to lower reperfusion injury in animal lung transplantational model in vivo. In a related role, thioredoxin is believed to be responsible for the development of cellular resistance to anti-cancer chemotherapeutic drugs like cis-diamine dichloroplatinium. 2. Possibly by scavenging intracellular toxic oxidants generated by this anti-cancer agent. Recent studies suggest that thiol transferase, also known as glutaredoxin, together with glutathione reductase and NADPH, also repair oxidatively damaged proteins. The evidence indicates that thioredoxin and glutaredoxin show different specificity in their interactions with protein substrates. Thioredoxin has also been found to act as an autocrine growth factor for both types of cells. T and B cell lines. Thioredoxin plays an as yet undefined role in DNA replication in mammalian cells. Thioredoxin can serve as a substrate for several enzymes, including methionine sulfoxide reductase, protein disulfide isomerase, a novel type of peroxide reductase, and as noted above, ribonucleotide reductase, although here glutathione reductase glutaredoxin appears to be a physiological cofactor. In addition to substrate function, thioredoxin reductively regulates different processes involving protein receptor interaction, toxin inactivation, or DNA binding activity of transcription factors whose activity is modulated by incoming signals to immunologically important transcription factors NF-κB and activator protein 1 are both activated by redox-dependent processes. Thioredoxin seems to play two different roles in the redox control of the NF-κB DNA interaction, one in the cytosol and the other in the nucleus. According to one group, thioredoxin appears to inhibit the release of IKB as one of the regulatory steps of NF-κB activation via pathway involving a reactive oxygen intermediate. Then free NF-κB would act in the nucleus to bind DNA. Other types of transcription factors are also under redox control by thioredoxin, including ones responsible for the stimulation of expression of cytokine genes. Thioredoxin may act as a competence factor to sensitize a cell and make it responsive to minimal amounts of a series of growth factors at certain steps of the virus transformation process. Thioredoxin thus has co-cytokine activity in enhancing growth of transforming lymphocytes and the expression of interleukin-2R. This effect is dependent on its disulfide reducing activity. Thioredoxin can promote B-cell differentiation. Endogenous thioredoxin expression is induced in both normal B lymphocytes and in leukemic B cells, chronic lymphocyte leukemia. After growth stimulation, these results suggest that induction of thioredoxin synthesis and secretion may be a normal stimulus of B lymphocyte activation and not necessarily linked to cellular malignancy. Recent data have shown that thioredoxin is able to induce critical early signals essential for the initiation of cell division. As an example, thioredoxin efficiently increases intracellular calcium and activates protein kinase C. Protein kinase C through its translocation to the membrane. One of the most exciting developments in the past 5 years has been demonstration that thioredoxin controls central function in the body defense system. There is, as mentioned above, evidence that thioredoxin function in the activation, differentiation, and proliferation of lymphocytes. Independent experiments suggest a role in enhancing the non-specific defense system through increasing the migration and cytotoxicity of eosinophils. It is well known that radiation of cells or tissue with UV, especially UVB, generates intracellular reactive oxygen intermediates, which subsequently induce various cytotoxic effects, including DNA damage and immune suppression. Thioredoxin secretion is enhanced in lymphoid cell as well as epidermal cells in sun-exposed skin. The results suggest that thioredoxin is induced by oxidative reactions and acts as anti-stress protein to repair cellular damage. The expression of thioredoxin messenger RNA and secretion of the protein are elevated in cancerous cells of the lung, the liver, the breast, as well as in those tumor cells that have been analyzed. Thioredoxin plant. Plants have 3 types of thioredoxin, whereas animals have only 1. The 2 chloroplast thioredoxin F and M have been intensively since their identification because of their importance in regulating photosynthetic activity. The 3rd class of plant thioredoxin, the H type, is located in the cytosol and endoplasmic reticulum. The thioredoxin, perhaps the H type, has also been described for mitochondria. Thioredoxin H differs in both structure and activity from the F and M type thioredoxin of chloroplast. The first evidence that the NADP thioredoxin system specifically reduces intramolecular disulfide bonds of small proteins was obtained. Thioredoxin was also found to reduce and thereby activate the new type of serine protease named thiocalcin. With endosperm, thiocalcin also requires calcium for activity. Thioredoxin in combination with calcium was the most effective reducing agent for the activation of the enzyme that had been oxidized in vitro. Several lines of evidence support the conclusion that thioredoxin promotes the mobilization of carbon and nitrogen in the early period of seedling. development during germination. Experiments show that the endosperm fraction contains the enzyme necessary to reduce NADP by the oxidative pentose phosphate pathway, hexokinase glucose 6-phosphate dehydrogenase, 6-phosphogluconate dehydrogenase. The abundance of thyrodoxine H in wheat endosperm appeared to be controlled by the embryo via the hormones gibberellic and abscisic acid. Thyrodoxine appears to promote the formation of protein network, improves the final baked product. Finally, the application of thyrodoxine to food such as wheat decreases their allergenity by mitigating the immune globulin E, mediates type 1 hypersensitivity response in sensitized dogs. Thyrodoxine reduced by NADPH and NTR. Thyrodoxine in medicine. Thyrodoxine are small proteins with the catalytically active disulfide group and that function in a spectrum of biochemical processes. Oxidized form of thyrodoxine contains a disulfide bridge that is reduced to the sulfitryl level by either reduced ferrodoxine or NADPH via one of two specific enzymes. The nature of reductants serves as a basis for distinguishing the two thyrodoxine systems. Reduced form of thyrodoxine is a very efficient catalyst for the reduction of disulfide bonds. The ferrodoxine-linked thyrodoxine system is located in oxygenic photosynthetic cells. It functions by linking light to the regulation of enzyme of carbon assimilation and related processes. It is composed of ferrodoxine, an iron-sulfur enzyme called ferrodoxine-thyrodoxine reductase. The NADPH-linked thyrodoxine system is composed of NADPH of flavin enzyme called NADP-thyrodoxine reductase, and as an associated thyrodoxine of yet another type, thyrodoxine reduced by NADPH has emerged as a regulatory protein of fundamental importance to plants as well as animals. Thyrodoxine linked to NADPH effectively and specifically reduce intramolecular intrachain disulfide bonds of an array of C proteins, including storage protein and enzyme inhibitors. Thiocalcin enzyme appeared to act independently of calmodulin. Thiocalcin appears to be representative of a new type of protease that depends on two well-known cellular agents, thyrodoxine and calcium. Certain proteases, calpain, are known to be activated by calcium, but none require thyrodoxine. The results demonstrate that thyrodoxine acting jointly with calcium functions to enhance proteolysis. through the activation of thiocalcin. In addition to its earlier identified function is reducing, solubilizing storage proteins and neutralizing enzyme inhibitor proteins. Thioredoxin animals, cell division, radical scavenging, non-specific defense, transcription, DNA replication, immune response, detoxification, meiosis, embryogenesis. The vastes stretched large are around the mine and were contaminated soil, plant, and water carcinogens were found in vast of the mine. Residents were exposed to these contaminants. Incidence of cancer and chronic disease would increase in the future. The vasted area was the focus of the environmental and health problems. The area is classified as the noticeable coastal ecosystem, but sadly the high amount of arsenic, barium, cadmium, chromium, and selenium in the environment are surprisingly noticeable in the aspect of human health problems, increased and expanded because the pollution in water, air, and soil affected. The biodiversity, water-soil balance was affected apart from agricultural production of olive, citrus, and carrots during 58 years of mining. And copper concentration plant activities, a huge waste material has been accumulated in the tailing ponds at Gemikonal. The rich pyrite content of the ponds has resulted in acidity in the pond water and sulfur contamination of the air. However, at present, sulfur pollution has been reduced and does not affect even nearby citrus plants. It is advisable to treat the plant content with lime to eliminate the acidity of the ponds. Cyprus, as it is known, surface and underground mining have caused environmental problems due to acidic discharge, commonly referred to as acid mine drainage. Here exposed sulfite minerals react with oxygen and water to produce an acidic solution that dissolves metals and contains sulfate. CMC and its waste, unsuitable for irrigation purposes and contamination resulting from the operations of the Cyprus Mining Corporation. This was a proof for water pollution and at the same time a potential cause for land pollution. Reduced productivity of the citrus orchards was defined in the report as follows. With regard to production, it has been visually estimated that the number of fruit per tree of both Valencia and Jaffa was less than half the number produced by trees of similar and undersized fruit of Valencia oranges was higher than that on normal trees. In other areas, contamination of the marine life and creating potential human health hazard as a result of fishing activities around this area. Furthermore, the moist sludge collected from the inside of the established facility at the same site was then analyzed and proved to contain high amount, 3-6 times more than the maximum concentration limits of arsenic and selenium, both of which are known to be hazardous to human health. The second site, having a number of large ponds, contains considerable amounts of copper porphyry. These tailing deposits are rich in sulfur, and when the contact with rain, water and oxygen, sulfuric acid is dangerous gas is formed, hence resulting in air pollution. Ponds in this area are potential sources of groundwater contamination. For the whole area, the analytical data for soil sample 2A showed high concentration of arsenic and selenium. The analysis showed concentrations of arsenic at 15.8 mg/liter and selenium at 6.39 mg/liter, which are above MCL of 5 mg/liter and 1 mg/liter respectively. Arsenic is a teratogen and carcinogen that can produce fatal death and malformation in animals. It is also a carcinogenic in humans. Many marine organisms and fish in the sea exhibit high concentrations of arsenic. Some species, such as lobsters, have as much as 100 parts per million. In general, arsenic may be absorbed by ingestion, inhalation, or through permeation of skin or mucous membranes. Thank you very much.